All right, everyone, welcome. Today we're going to jump right into a really central concept in biochem, something called the amino acid pool. You can think of it as the ultimate currency exchange for just about everything protein-related happening inside your body. So here's what we've got on the agenda. First, what is this pool? Then we'll look at how it gets filled up, the deposits, and how it gets used, the withdrawals. We'll also tackle the big question of what happens when you have too many amino acids. And finally, we'll bring it all together and see why this is so critical in a clinical setting. All right, first things first, let's really define what we mean when we say the amino acid pool. Okay, now the first thing to get straight is this. It's not a literal pool, like a swimming pool filled with amino acids. A much better way to think about it is like a central bank account. You've got all these free amino acids floating around in your body's fluids and tissues, and this pool is just the total collection of them, ready for deposits and withdrawals at any moment. And if you were to add it all up, how big is this account? Well, it's surprisingly small. In a typical adult, we're only talking about 100 grams of free amino acids. And here's what's really wild. About half of that, roughly 50 grams, is made up of just two amino acids, glutamate and glutamine. So yeah, they are the absolute major players in this whole system. So that leads to a pretty obvious question, right? If the pool is only 100 grams, how does your body keep it topped up? Well, there are three main ways you make deposits into this amino acid account. Let's break them down. Okay, source number one is something called body protein turnover. This is basically your body's own recycling program. It's constantly breaking down old or damaged proteins and just tossing those amino acids right back into the pool to be used again. The second one is the most straightforward. It's the protein you eat. Simple as that. And third, your body is a pretty amazing little chemist. It can actually make its own non-essential amino acids from scratch. Now check out this number because it's pretty mind-blowing. Every single day, your body is turning over somewhere between 300 and 400 grams of protein. Just think about that for a second. The pool itself is only 100 grams, but the turnover is 3 to 4 times bigger. That just goes to show you how incredibly active and dynamic this whole system really is. Okay, so we've covered the deposits. Now let's look at the withdrawals. Where are all these amino acids going? What are they being used for? Well, no surprise here, the biggest withdrawal by far is from making new proteins. That 300 to 400 grams we just talked about, most of it gets pulled right back out to build new muscles, enzymes, you name it. Then, a smaller chunk, maybe about 30 grams a day, has a very special job. It's used to make other super important things that contain nitrogen, like the purines needed for your DNA. And whatever is left over, the body is resourceful. It can burn it for energy or even convert it into glucose or fat if it needs to. Okay, but what if there's a surplus? What if you have too many amino acids in the pool? Now, this is a really, really important point. Unlike fat or glucose, which we can store pretty easily, your body has absolutely no storage facility for extra amino acids. None. So they have to be broken down. And that creates a problem, a nitrogen problem. So the first step in dealing with this is a super clever process called transamination. The hot potato analogy is perfect here. The body takes the amino group, that's the part with the nitrogen, from one amino acid and just passes it over to another molecule called a keto acid. It's a simple swap. And a key detail here, this reaction is totally reversible, and it absolutely needs vitamin B6 to work. That's a classic exam question, so make a mental note of that. All right, then comes the next step, deamination. And this is a whole different ballgame. Deamination literally means removing the amino group. It's not being passed along anymore, it's being ripped off completely. And when that happens, it's released as free ammonia. And that, my friends, is a big problem. Ammonia is toxic, so the body has to get rid of it, and fast. So let's just nail down this difference because it's crucial. Transamination is all about shuffling nitrogen. It's moving it around inside the body. Deamination, on the other hand, is about removing nitrogen. It's the first step to kicking that nitrogen out of the body for good. So how does the body do this efficiently? Well, it's pretty smart. Through a whole bunch of those transamination reactions, nitrogen from all sorts of different amino acids gets funneled onto one main molecule, glutamate. Think of glutamate as the central nitrogen collection center. Once the nitrogen is on glutamate, that's the molecule that finally undergoes deamination to release the ammonia. Right, so now we've got this free ammonia floating around. And as I said, this is a big deal. This stuff is seriously toxic. and your brain is particularly sensitive to it. And this quote from Satyanarayana says it all. You don't need a huge spike in ammonia to cause damage. Even a tiny bit extra can be harmful to the brain. This really underscores why the body needs a robust, efficient way to neutralize and excrete it immediately. So, what's the solution? It's a beautiful biochemical pathway called the urea cycle. This whole process goes down in the liver. Its entire purpose is to take that toxic ammonia and convert it into something totally harmless and easy to get rid of. Urea. Urea is water-soluble, so it can just dissolve in the blood, travel to the kidneys, and be excreted. But look at the energy cost. This process isn't free. It costs the body 4 ATP molecules to make 1 molecule of urea. It's a price the body is willing to pay to stay safe. Okay, so let's shift gears. We've done the biochem. Now let's make this clinical. Why do you, as a future doctor, need to know all of this? How does it actually matter when you're seeing patients? It matters because we can measure the final product of this whole pathway. You can check the level of urea in a patient's blood. You're going to hear the term BUN, or blood urea nitrogen, 1,000 times in your career. It's a standard blood test, and it gives you a fantastic window into 2 big things: how well the kidneys are working and what's going on with protein metabolism. Now, if you get a lab result back and the blood urea is high, that's a condition called uremia. Your first thought might be, oh no, kidney failure. But hold on. A high BUN level makes you a clinical detective. You have to ask, why is it high? Is the problem happening before it even gets to the kidney? We call that pre-renal. Or is the problem in the kidney itself? That's renal. Or is there a blockage after the kidney? That's post-renal. This pre-renal, renal, and post-renal framework is something you will use constantly. So let's bring it all home. I want you to leave today really thinking of the amino acid pool as this dynamic, central hub for metabolism. Everything has to be in balance: the deposits, the withdrawals, and the waste disposal. When that balance gets thrown off, you get disease. And understanding how it gets thrown off is your key to diagnosing so many different conditions, from kidney disease to inherited metabolic disorders.